I'm Lénia Rato from Maré Polytechnic of Leiria in Portugal. So Maré is this very comprehensive marine research institute that is spread around our country. In the center where I work in Peniche, research is developed mostly towards aquaculture, marine ecology, and also blue biotech. So my work relates mostly to marine ecology. In the past few years, I've been researching how species respond to environmental stress and also climate change. And I've been doing it from crustaceans to fish to invasive jellyfish and addressing responses at several levels of biological organization. So we wanted to gather the knowledge that I've been gathering these past few years and design this PhD thesis that I've been working, who assesses both climate change and bioevasions and see how they interact with each other. So I target for crab species, namely Emigraptus stachanoi, that is spreading in the north of Europe, and also Crescinus mayanus, that is our native species. And we see how they respond to the same stressors at the same time in the same experimental setup, which allows us to compare their responses and try to understand their plasticity and if there are any competitive advantages across species. So in the past few months, I've been studying how this species responds to pollution, namely in the form of a pesticide that is used as an alternative to neonicotinoids that we know that are very toxic to pollinators. We hypothesized that this new generation pesticide could have detrimental effects on this non-target species. We assessed function, behavior, and also physiology in both species. So for the behavior, we assessed how long would they take to reach the food, and then for their function, how much would they eat. As a physiological proxy, respiration rates were measured thanks to Pyroscience sponsorship. So we had this really cool setup with optical reading and sensor spots that allowed to minimize handling and manipulation stress in the organisms and obtain really nice readings on respirometry. The results suggest that both species were affected by the presence of the pesticide, but the invasive one was more impacted since it had differences in all the proxies that were addressed. I wanted to thank all the funding and sponsoring companies and agencies who support our research. Once again, Parascience for their help regarding this equipment and their sponsorship under the ICMARE conference.